Hi, welcome to HESI Test Practice Lesson 3 about 10 important biochemistry-related question which has encountered in HESI test. Today our first problem will be, question number 1. Which of the following is the primary function of enzymes in biochemical reactions? A. Increase the rate of reaction by lowering activation energy. B. Decrease the rate of reaction. C. Provide energy for reactions. D. Act as a substrate for reactions. Answer is A. Increase the rate of reaction by lowering activation energy. Question number 2. What is the main energy currency of the cell? A. ATP, adenosine triphosphate. B. NADH, nicotinamide adenine dinucleotide. C. GTP, guanosine triphosphate. D. FADH2, flavin adenine dinucleotide. Answer is A. ATP, adenosine triphosphate. Question number 3. Which of the following is the source of energy in muscle cells during short bursts of intense activity? A. Fats. B. Glucose, glycogen. C. Protein. D. Oxygen. Answer is B. Glucose, glycogen. Question 4. What is the function of the ribosome in a cell? A. DNA replication. B. Protein synthesis. C. Lipid synthesis. D. Energy production. Answer is B. Protein synthesis. Question 5. Which of the following is a component of the plasma membrane of a cell? A. DNA. B. Phospholipids. C. Glycogen. D. Proteins. Answer is B. Phospholipids. Question 6. Which of the following hormones is involved in the regulation of glucose metabolism? A. Insulin. B. Estrogen. C. Thyroxine. D. Cortisol. Answer is A. Insulin. 7. What is the first step in the glycolysis pathway? A. Formation of glucose 6-phosphate. B. Conversion of glucose to pyruvate. C. Phosphorylation of fructose 6-phosphate. D. Hydrolysis of ADP. Correct answer is A. This reaction is catalyzed by the enzyme hexokinase, or glucosidase in the liver, and requires ADP. Reaction, glucose plus ADP, glucose 6-phosphate plus ADP. This step traps glucose inside the cell and prepares it for further breakdown in glycolysis. Question 8. Which enzyme catalyzes the conversion of glucose to glucose 6-phosphate in glycolysis? A. Hexokinase. B. Phosphofructokinase. C. Pyruvate kinase. D. Aldolase. Answer is A. Hexokinase. Explanation: this reaction requires ADP, which donates a phosphate group to glucose, preventing it from leaving the cell. Reaction: glucose plus ADP, hexokinase glucose 6-phosphate plus ADP. In the liver, a similar enzyme called glucosidase performs the same function but with a higher specificity for glucose. Which molecule is produced at the end of the glycolysis process? A. NAD. B. Acetyl-CoA. C. Pyruvate. D. Glucose. Answer is C. Pyruvate. Explanation: glycolysis occurs in the cytoplasm and consists of 10 enzymatic steps, ultimately breaking down 1-glucose, C6H2O6, into 2-pyruvate, C3H4O3, molecules, along with energy-rich molecules ADP and NADH. Reaction summary: glucose plus 2-ADP plus 2-pi plus 2-NAD plus 2-pyruvate plus 2-ADP plus 2-NADH plus 2H2O. 10. How many ADP molecules are consumed in the early stages of glycolysis? A. 1. B. 2. C. 3. D. 4. Correct answer, B. 2. Explanation: in the early stages of glycolysis, the energy investment phase, 2 ADP molecules are consumed to phosphorylate glucose and prepare it for breakdown. Steps where ADP is consumed: 1. Glucose 6-phosphate, G6P. Enzyme, hexokinase. ADP used: 1. 2-fructose 6-phosphate fructose 1, 6-bisphosphate, F1, 6BP. Enzyme, phosphofructokinase, PFK. ADP used: 1.